Who start? You have to speak in Italian or in English? In Italiano?
Interpretation is provided both ways, so
are there any questions
in English or in Italian? Feel free to ask questions either in English. Feel free to ask questions in English or in Italian. Interpretation is provided both ways. And of course, you're free to answer in your own language.
Allora.
Here we are. My question is quite straightforward as a parent. It's something that has to do with what Alessandro said in closing, i.e. the possibility of correcting through gene therapy brains that are already formed. Is this a possibility? I mean, obviously, this is any parent's hope, because many of us have girls whose brain development has ended, and we wonder if there is hope, for brains that have already developed.
There's also a session on that that we're having tomorrow, but I guess that's just the scientists, right? So I would think that it would be easier to suppress the wild type, the normal gene function, to make it completely absent. And I would think that, so there are ways to do that with something called CRISPR interference, but my bet is ASOs would be quicker and maybe an easier initial approach to do that and see if it works. Antisense oligonucleotide therapy, which is already in clinical use, because it doesn't matter if you hit the mutant or the wild type allele if you're going to knock it down.
Okay.
Probably have other people that want
I would agree. What we really learned from other disorders, neurological disorders, that this is feasible, and there are probably hundreds of disorders and groups around the world who are exploring antisense therapy with delivery to the brain for these purposes. In this case, as Jack alluded to, we are probably fortunate that we can afford to destroy even what is working, but that would need really good studies in the preclinical model, of course, before we go into. My, I guess, question is, and again, it's a preclinical study, what part of the problem is actually cell autonomous? So if this is really the terminal function of the molecule to sit somewhere around the synapses, how much by correcting that we can return it to normality. But even if we can return it by 20% or 10%, it's going to be fantastic, and it's going to be worthwhile doing it. Now, that's what, as parents and as scientists, we need to realize, that it's going to be almost not possible to bring it back to normal as other average population. But if we improve it by the antisense therapy, even at the age of 5 years, by 10 or 20%, that we improve the behavior, we get rid of the seizures, it's going to be a win. So I think we need to be modest in our expectations, but persistent with our trials.
Io ho una domanda. Put your...
I have a question. You need to wear your headset because I can only speak in Italian. Apologies. So, I am not an expert in the field of PCDH19, Joseph. You know me, but I do work on other neurodevelopmental disorders. The question we're all asking is treatment possibilities. What are they? There are many genes, and that's a problem, that can't be targeted directly. We cannot modify certain genes directly when we're talking about brain neurons, that is. However, there are talks about different penetrants, siblings with the same mutation and different clinical features, modified gene expression, etc. I wonder whether any of you is currently exploring the following option. I did some research, but I might have missed something. Are you considering the following option? Is any of you doing RNA-seq or sequencing or gene expression profiling? And if so, where would we have to do it? That is a specific question for PCDH19, but members of scientific communities like myself really would need to know this to see what a possible approach for other diseases could possibly be. Who is going to answer? Who could answer this question? Joseph.
I will have a quick comment and I will let others. So the question here, as I understand, Patricia is asking, when we do the treatment, do we actually treat the PCDH19 or do we also focus on other consequences? Because it is kind of like a pyramid upside down. We start with PCDH19, which is not working properly, but consequently, as the organism grows, there's other consequences. So as I showed you, the ball ends up in a different landscape, so it has a different characteristics. So fundamentally, we can target the molecule by CRISPR or antisense oligonucleotides, or as we also invested a lot of work, and we heard from Alex, we try to identify the consequences, pick the most important consequences by biomarkers, and we target those. And in this case, we noticed that these girls are deficient in allopregnanolone. Ganaxolone was the obvious, naturally available substitute. So technically, we're doing a hormone replacement therapy. But there might be other molecules, there might be other similar pathways, where there can be a diet, it can be other interventions which we should be keeping in our horizon when we're looking at treating these girls, not just focusing on PCDH19 itself, but also on the other peripheral consequences.
So we're gearing up to do RNA-seq both in the mice and in the iPS-derived organoids, with the idea that if we could find other pathways to target with drugs. I think it would be nice to do a high-throughput screen. My friend and colleague, Scott Baraban, made the PCDH19 knockout fish model, but did not see a phenotype. So that would have been ideal for screening. But I think we need to keep in mind high-throughput screens. Yeah.
Maria?
You need to use a microphone, everyone, please. Yes, we can't hear any comments unless they're spoken into a microphone. We are working on RNA-seq on single cells, on mouse models and DPS models, cells as well.
C'è una domanda? Okay.
Question over there.
A comment and a question. A comment towards the therapy of knocking down protocadherin 19. So I think in that sense, the advantage of this disease is that you can go both ways. Because in theory, restoring the function in the knockout cells or removing it from the wild-type cells should give you a good outcome. But I think we should not forget that when we look at the transmitting males, as Joseph put them, we should not forget that these males have been lacking protocadherin 19 from the beginning. And as Joseph also pointed out, that's not the same situation as having had protocadherin 19 and then losing it acutely. So I think that there will be a lot of pre-studies to be done to see how it affects how having a mosaic system, whether you can really revert it, whether neurons are capable of rewiring, or how important is the wiring between neurons to the phenotype, apart from other actions on how excitable the cells are, etc. And then I have a specific question for, let me check the name, sorry. There we go. For Stefka. I was very impressed by your study where you take this single cell and you either have a knockout cell in a wild-type environment or vice versa. But then thinking about what is the situation in the brain, that's not really a very physiological situation, because you very rarely are going to have an isolated neuron that is either wild-type or knockout in a background of all the other ones. So can you comment on what you would expect the neurons to look like in terms of all these differences that you found in the setting of a true mosaic and not just one cell in a homogeneous background?
Works. All right. So the reason why we have tested it in this system is to see what is happening in the borderline between in the mosaic when we have the cells that express PCDH19 and that do not express PCDH19. But you are right that in the whole brain, that represents very small population of cells that are going to experience that changes. But our main hypothesis is actually these minor changes that can be seen in the borderline affect the neighbor cells. Because we performed some RT-qPCR analysis of the whole brain of the heterozygote females. And we've seen that actually the synaptic proteins like the synapsin-1 and synaptic adhesion molecules are increasing, which means that the improper interaction between the knockout and the wild-type cell, what we are seeing is just an isolated situation, could be extrapolated in the whole brain that somehow increasing the synaptic transmission in the rest of the cells because they previously didn't receive the proper signal of this interaction between the no and the wild-type cells. So actually our idea was to see if there is some alterations between the knockout and the wild-type cells. Probably that could be extrapolated like the mixing or just disrupting the network activity in general, the cells that are sitting close to this just border cell line. And the other thing that we are also planning to perform, and that will be done with our PCDH19, the conditional PCDH19 flux mouse. We're going to use our PCDH19 TAC and this PCDH19 flux mouse. So once the CRE-mediated recombinase occurs, we will know which cells are becoming green, that they will be knockout, and we can get the just brain slices and measure them by the multi-electrode arrays, how the electrophysiology of the whole slice works, and we will be able to identify which one are the knockout cells and how based on the electrode and where they are collecting the signal, seeing how in the whole batch of the knockout cells actually is altered the electrophysiological activities, what is happening in the borderline, even we should be very lucky to get the electrode sitting over there, and the wild type region. So we expect to see big differences in this context.
A comment? Isabel, I just want to address your first comment. And I think you're absolutely right that you have to worry about existing compensation in already functioning networks when you knock it down or knock it out. On the other hand, I worry about the balance. So if you're going to do CRISPR activation to try to increase expression of the wild type, or actually to turn on the mutant allele maybe, or I'm sorry, the inactivated wild type allele. I worry about the balance, and it could be that if you double it in some cells and bring it to normal in other cells, that you still might have a problem. There's a Cadherin literature that suggests overexpressing versus normal can interfere with the interaction.
Yeah, Rett syndrome, for example, of that situation, which is almost worse, to have overexpression than the lack of the gene. I did it tomorrow, will show data in the conditional model that if you lose PCDH19 in young adult, so after 60 days of age, you actually have a phenotype. And so that is well beyond after synaptogenesis, is after layering, of course. And so I think there are still very important indications. We actually need the protein, or we need both of them at that time. So I think we can go back to that tomorrow. Yeah. It's a milder phenotype, but is unquestionably there. And it looks like the phenotype you have when the loss is during early postnatal life, but it's simply milder. So I think that is something we should keep in mind.
C'era una domanda? Sì, là.
That might be good news.
I have a question. Is it possible for a subject to be born without the genetic mutation, and that the genetic mutation is then found only later on, that is after he has had the onset of the disease through a seizure? For example, that's the case of the girl that after 5 months developed, and I'm referring to external factors probably. For example, after 5 months she was having her vaccine, her second vaccine, and after that she manifested a seizure, the first one. Now, what if a subject is born with a mutation already, or, and in that case it's quite obvious that as soon as she's born or the baby is born, you do not conduct the investigation to try and ascertain whether there is a mutation. You only do that whenever you have a first seizure and you find out that there is this kind of condition. Is it possible that a subject is born healthy and you find it out, I mean, the condition is found only later on, in a later stage? Is it possible?
Posso rispondere io?
If you want, I may answer. Now, that a subject is born without a mutation, yes, that's possible, and it's males with a mosaic. The mutation onset takes place in some cells almost immediately after birth. This was not the case, not your case, because with females the mutation is present ever since birth. That is, ever since the zygote cell is formed, where we have the mutated allele and a normal allele. That mutation, since the cell is just one and then continues to divide and until when it's forming a whole organism. So with females, the answer is yes, mutation is present ever since the first cell exists. And based on what we heard this morning, it is also possible to have some trigger factors, such as fever or fever attacks, which may trigger the symptoms. So there is a genetic base which is predisposing to the disease, and then after that the disease becomes frank, and we know that the trigger effect is the one that triggers the disease from that moment onwards, or at least the symptoms of that disease. Because the condition is pre-existent, it's all only hidden until when there is this first episode from the genetic viewpoint. That means that it's always there ever since the beginning.
Add that you can be born with a gene mutation, even a somatic mutation, and not, you can be born with a gene mutation, germline or somatic, and not have seizures for 10, 20 years. So it can definitely be delayed.
The other potential complication is, of course, you can detect generally these mutations well before the individual gets seizures. So we have the technology. It is just expensive, and we don't do it because we don't have always good reasons to do it. But technically, we could test every newborn baby to see whether they have or don't have. They already do it for many conditions using newborn screening programs. Phenylketonuria is one example where they can prevent damage to the brain by testing early and doing proper diet. But there are also conditions in seizures where you don't detect the mutations in the blood. They can be only restricted to the brain tissue, and that's complicated. And we have not seen a case like that with PCDH19, but we've seen many cases like that with different seizure disorders, that we cannot detect the blood, the mutation in the blood or other part of the body. It's usually detected only after that part of the brain is surgically removed, and then people look at that part of the brain, they find the genetic cause of that problem. Now, we have not seen anything like that for PCDH19 epilepsy, but in principle, it also can happen, and we need to keep it in mind.
Salve, scusate, vorrei fare una piccola precisazione.
Good afternoon. Let me make a little comment. The adult subjects in a menopause state, may they have an altered organization, or may they have a temporal history? And could you specify any such aspects?
Aspetti, C'è qualche chimico che ci vuole aiutare? Yeah.
As I understand, also for everyone here, is whether we have enough data about the older women at the age of the menopause, and how they compare hormonally and clinically to the younger individuals. Now, we have only literally a handful of these individuals where we have seen restart of the seizures. We have two such older ladies who are from these pedigrees we found the PCDH19 in. These ladies have considerable learning disability and neuropsychiatric problems. They just were not having seizures. They are PCDH19 positive. We have not tested their hormonal levels, but as soon as they went into menopause, they restarted to have serious seizures. But that's, again, it may or may not be related to PCDH19. We can only speculate because we don't have numbers. Generally, menopause is quite a dramatic event in terms of hormonal balance, and we know it from catamenial epilepsy and others that hormones play a very important role in epileptogenesis.
This is for Silvia Bassani. My question. I don't know whether I understood all of the things you said. I'm not sure. I didn't have the headsets. But you see a dysfunction of the GABAergic system, which is due to the absence of PCDH19, because you did prove that there is an interaction. Until then, so far, so good. But now, the concept is that what you show in the cells is also visible on the mouse, on the slices. And this is a question that I think someone already asked this morning. Is it possible to start thinking that we might be using an agonist, antagonist GABA according to the phenotype? You probably have to use probably an agonist in this case to try and see if you have the rescue of the phenotype. Well, yes, definitely this is something which is worth trying. After that, as Jimmy was rightly saying, we still have to understand a whole series of factors. In this case, for example, we have a reduction of the GABAergic expression. We are to understand if there also is an altered switch at the GABA level. It might very well be. But definitely one of the things which may be tried is the one of trying to increase the GABAergic transmission and try and see if we can achieve something. Mosaicism are turning the whole thing into something difficult because we have to see whether we can correct the defect in a homogeneous manner. But definitely, yes, this is something we have in mind doing.
Tonic inhibition results relate to the neurosteroid issues and potential mechanism?
Well, yes, I think that we see the protocadherin effect on the GABAergic currents and on the tonic currents, and we were wondering why. There are a number of different mechanisms. So one could be due to the physical interaction, because we well know that protein-to-protein interaction may change the transmission of the channels. But then there could also be many explanations and indirect mechanism. So we know that there seems to be an indirect effect. And one effect was exactly the one of increasing the opening time of that channel. And so that could be sort of a way to support, so that our data could support the positive effect generated by ganaxolone.
Even after your talk, as I understand, your model is non-mosaic. So these are all homogeneous cells.
Well, yes, as far as the slices, the brain mouse slices, it's very similar to a mosaic in the sense that we do not have an expression or a modulation, rather, within all of the cells of that slice, because we cannot make it 100%. And then probably we are now using another model where there is no knockdown, but rather there is going to be a knockout. But that's a mosaic.
That will be a mosaic, because not all cells will be hit, but you will generate different mosaicism because there is not going to be in clusters. But your cell type model is uniform, yet you see a difference. So by taking it comparison to the transmitting males, the loss of PCDH19 should not cause a problem. So what you see, it's in fact another normal situation. So it's not a pathological pathogen.
So whenever we do experiments in culture, and we are obviously looking at the characteristic affecting each individual cell. After that, as I said before, at the end of the presentation, what we would like to do is to extend all this at mosaic level, because this kind of heterogeneous model could provoke some GABAergic problems at circuitry level. As far as the boys or the males are concerned, we should also say that if this is a homogeneous defect, it might very well be that there are some homeostatic mechanisms which are more than sufficient if the defect has been enrolled in a different way. And we might have then the more complex homeostasis mechanism, which still have to be proven.
We made from males, because we have a male, iPS and neurons. They behave differently to a wild-type neurons. Yet it's another normal situation, because we know these males we derive these cells from are not affected. So what we have seen was premature differentiation and early premature migration. That was the hallmark of the study. So I guess we're learning all new biology, that we have not one normal, we have multiple normals. And this disorder is a clash of two normals. Yeah. And that's a fundamentally very different concept than what we so far have been studying in this space.
Noi dobbiamo conoscere anzitutto molto bene I meccanismi di compensazione.
We have to get excellent knowledge of compensation mechanisms that come into the play, especially with the males, the knockout males, because that is what may account for the reason why a phenotype is not manifested, and how comes that this is not happening whenever there is a mixed population. At the end, what is explaining or determining our behavior is not the property of each individual neuron, but rather the circuitry and the population. And the setup of the circuitry is very much robust, and it resists insults and compensations. So reducing the GABA in all the neurons, allowing for the efficiency of the GABA, is what leads to a compensation on the excitatory state at all neuron level. But if you only have a fraction of neurons, then the final result is going to be absolutely different. And then maybe, I say maybe, this is what is at the very basis of these mysteries. So the fact of a mosaicism that could be very well due to the cell outcoming effect.
Buonasera.
I'm the mother of a girl with the syndrome, aged 4. I would like to know if there are any predisposing factors which bring about this disease. Now, could you please tell me what do you mean by predisposing factors? What are these predisposing factors? Well, when I was young, I had epilepsy, so I would like to know whether there is an inheritance. But this is a de novo disease, so my daughter is not the result or the outcome of me. Is there anything that favors the onset of this disease? This is what I want to know.
It's not a simple answer to your question, but in some cases, like if your daughter has a genetic mutation which explains her seizures, sometimes this genetic mutation is also found in parents, but in a very small proportion of cells, which usually it's not detectable using standard tests. So you need to go to very special tests, which very few people do. So in a way, this is, again, a consequence of so-called somatic mosaicism. So you may have a seizure because you may have that particular genetic alterations in a very specific part of your brain, but not necessarily in the blood. But you passed it on through your gametes, through your eggs, to your offspring, and they have it in every single cell. So they may have a full-blown genetic epilepsy, while you might be only very, very little affected because you have this particular genetic predisposition only, let's say, 2% of your cells. It's enough to cause a seizure or two, but not to give the full-blown disorder. That's a possibility from experience in epilepsies, which is now well demonstrated to exist. Of course, there can be other explanations, not related to this. This is quite special. That might be other explanation related to polygenic risk or the background risk or other events which may not be or could not be linked to a single gene or a single piece of DNA.
Jack, do you
Chiedo
want
scusa.
I would just say, or you could have epilepsy from a totally different reason, so.
Nel mio caso invece
In my case, can you hear me? In my case, the only thing that happened in the mother and in the daughter is the CHRN4 mutation, and they told me that that was non-relevant, so they never dig into that factor. So I would like to know whether maybe you know what are we talking about, the CHRN4? Is there a correlation with the CHRN4 with the PCDH19? Is there a correlation in between the two things? No.
Nicotinic acetylcholine receptor, this is the first epilepsy gene which have been identified. I don't know that there would be any link between that and PCDH19, but maybe that gene variant on its own would explain the seizure disorder.
Did they tell you it was a pathologic mutation?
No.
No. They told me that that was a de novo mutation affecting the child, but what they saw was the CHRN4, and I had absences when I was young. In between the age of 6 and 10, I always thought that that problem could be depending from me. A month ago, I received the diagnosis, and that was really a hard hit because I had a positive evolution, and I was hoping that that was going to be the same with my daughter, which instead was not the case, because what I got was the PCDH19, which is totally different. Now, just to clarify, I think I can recall the genetic response of the genetic text, and I think it was the variant of CHRN4, which was a variant, quite significant one, to which we did not attach any importance because the phenotype was instead typical of the PCDH19. I think that this is the only answer I can give you. We probably will have to study mosaicism or CHRN4.
Col CHRN4. Sì, anche se di CHRN4.
The CHRN4 is present with different mutations. This is a recurring variant, a frequently seen one, and the mother did not have a picture which was a typical one for frontal epilepsy, just like the phenotype. So it was slightly a different condition. We know that there are hundreds of genetic mutations, and not only PCDH19. Are there any cases where we could sort of restore, if you pass me the term and the word, or solve? Maybe solve is the right word, he says. When we say modified, we intend it in the positive way. And if yes, what was the time period in between the discovery and the repairing moment? Or we talk about gene replacement in a number of conditions, such as cystic fibrosis. This is not a neurologic condition, so we do not have a problem of having to reach the brain and see what is the precise timing. But yes, there is a possibility, a successful one, where we could have some gene replacement, especially with metabolic diseases. So diseases where there is epilepsy, and in general, genetic mutation, which is not the PCDH19, because as of today this is not the case, but any other mutation which was solved and repaired. Now, as far as I know, is that there are some diseases affecting the central nervous system which are being solved thanks to gene therapy. a number of conditions. Metachromatic leukodystrophy, for example, is one. But there we talk about the genes having a enzymatic function. So in that case, seems to be an easier case. But that's not the same thing of a mutation. Well, these genes have already are already mutated. That's the function of the gene. And when the gene performs an enzymatic function, it's enough to restore one copy, a normal copy of the gene, so as to allow for that gene translated into the protein will go and perform his own function. It's easier for those genes encoding those enzymatic genes. So could we have a restoration, 100% restoration, or is there a percentage whereby we could be happy to have some 10-15% improvement? Now, the cases that you just quoted have been completely solved. For the ADASKID, I can tell you that since they do it within the San Raffaele Institute, we know that that's been solved 100%, but you have to treat them just like the metachromatic leukodystrophy, and the children have to be in their very early ages. As when they grow older, they don't even treat them, but it works very well. There are children who are just doing fine. I've seen them.
Though, you don't have to hit every cell because they can be secreted and get other cells. People have been successful with gene therapy in the retina, but that's easier to address. In terms of the brain, you're right to say we're unlikely to get it in all the cells. So the question is what cells where and what percentage do we need to hit to make things better, and we need to do the work to figure that out.
But another good example is hemophilia. You know hemophilia, the chronic bleeding disorder. These individuals need to get the clotting factor every month or 2 months. It costs millions of dollars of a lifetime. Hemophilia B has been successfully treated by gene therapy. They receive 1 dose of corrected cells. They don't need to go back for clotting factor ever again. It is working. We probably still early days. The patients need to be monitored how well the gene correction is working, whether there are not any side effects. But there is a reason for optimism. We have examples from these peripheral disorders, muscular or blood hematological disorders where it's working. Probably sickle cell anemia might be the next one, which is big on the list. I'm sure neurological disorders will follow sooner or later.